Okay, welcome everyone to today's Myeloma UK CARP webinar. I think we're probably still a few people to join. I know usually we get a bit of an influx over the first few minutes, so just while we're waiting, just want to let you all know that we will record today's session so that we can make it available on our CARP YouTube channel for those who want to catch up again later. Okay, Tyrion, have we got anyone else waiting to be let in, or shall we make a start?
No, I think we're ready to make a start.
Great, thank you. Okay, so as I said, welcome everyone to today's CARP webinar. So today I would like to introduce Dr. Holger Auner. He's a clinical reader in molecular oncology in the Centre for Haematology in Imperial London. He's also the translational lead on the UKMRA senior leadership team. We've worked closely with Holger on the MUKeight trial, which he's going to talk to us about today. But he's also working closely with us within CARP on the development of our translational database interface. So some of you who work within CARP at the CTIU may well have had some interaction with Holger before. So I'm delighted to welcome him to this webinar to present the results of the MUKeight trial, which is looking at the use of ixazomib in combination with cyclophosphamide and dexamethasone for patients with relapsed refractory myeloma. So I would like to hand over to Holger for our presentation. We will take questions at the end, if that's all right with everybody. So we'll come to that shortly. So over to you, Holger.
Thank you very much, Sarah, for the introduction. Please shout if suddenly the connection failed. As you can see, I'm in my kitchen today. I had no other choice but doing it. Hopefully the internet will continue to work fine. Thank you everybody for tuning in. I know it's a beautiful afternoon in most parts of the country, so I promise not to drag this out for too long. And I'll just make a start, and hopefully you will see the slide moving on. So as Sarah already said, what I'm going to talk about is the MUKeight trial results. And I would like to start by saying a huge thank you to the entire team that worked on this, and the planning, the setup, the execution, all the centers that took part, our colleagues, the nurses, the patients. And this study was very recently published in Blood Cancer Journal, so you can also look up details or email me if you have any questions or requests. So this was a phase 2 randomized controlled trial. I'd like to briefly start with a rationale for doing an ixacyclodex or ICD trial. As you will know, ixazomib is an oral small molecule proteasome inhibitor. It was approved by the US FDA and the EMA in combination with lenalidomide and dexamethasone in patients with at least one prior line of therapy based on the well-known phase 3 tourmaline MM1 trial. And what you can see here is probably the main result from this trial that you will have seen many times, the progression-free survival in the intention to treat population, where IRD had a PFS of just over 20 months compared to 14.7 months for RD. Also, there have been promising results of ixa combinations with pomalidomide and thalidomide, so it seems like a combination of an IMiD with ixazomib is effective. And furthermore, the rationale for using ixacyclodex is that IMiDs are not maybe always ideal for subsets of patients, for example, those with severe renal impairment, those with increased risk of thromboembolic complications, those who have progressed on an IMiD. So based on other combinations of a proteasome inhibitor with an alkylating agent and steroids like VCD, it seemed that a regimen of ixazomib with dexamethasone and cyclophosphamide could be a potentially quite attractive all-oral 3-drug approach. Also, and these are the figures I'm showing here on this slide, ICD had been shown to be effective and quite convenient with a reasonably favorable toxicity profile in both relapsed refractory and newly diagnosed multiple myeloma. On the left-hand side of the screen, you can see the main result from a phase 2 study of ICD in relapsed refractory myeloma, 78 patients, 63 years old, 1 to 3 prior lines of therapy, with an overall response rate of just under 50% and a median PFS of 14 months. On the right-hand side, you can see the findings in newly diagnosed patients, 70 patients, 73 years old, so these were transplant non-eligible, who received 13 cycles of ICD followed by ixa maintenance, with, as you would expect in newly diagnosed patients, a much higher response rate of 73% and a median PFS of 23 months across slightly different dosing arms. So this then resulted in the design of MUKeight, a randomized, controlled, parallel group, open, multicenter phase 2 trial, the primary objective of which was to evaluate whether ICD has improved clinical activity compared to CD, cyclodex, in terms of progression-free survival. In patients with RRMM who have relapsed after treatment with thalidomide, lenalidomide, and a proteasome inhibitor. And the reason why it was defined like this was simply based on what was available and what was routine use in the NHS based on NICE recommendations about 5-6 years ago. There were also a number of secondary and exploratory objectives, which I won't go into too much, like to determine the safety and toxicity profile, cost effectiveness, quality of life, in the paper if you want to look it up. So the inclusion criteria were pretty standard for a multiple myeloma trial, and I've highlighted the one that really makes it stand out. As I already said, relapsed refractory myeloma requiring further treatment following exposure to thal, len, and a proteasome inhibitor, regardless of the response to these. Exclusion criteria were also pretty standard. Now, what was the regimen like of ICD in MUKeight, and what were the standard dose modifications? The starting dose for all patients was generally ixazomib 4 milligrams on days 1, 8, and 15. Cyclophosphamide 500 milligrams days 1, 8, and 15, and dexamethasone 40 milligrams on days 1 to 4 and 12 to 15, or 20 milligrams on those days for older or less fit participants. Those reductions were written into the protocol. Ixazomib was reduced to 3, then to 2.3 milligrams on days 1, 8, and 5, cyclophosphamide to 400, 300, and 200 milligrams, and dexamethasone to 20 milligrams, 10 milligrams, or 10 milligrams on half the amount of days of the original planned. So this is the concert flow diagram, and I realize it's a bit small. Hopefully you can see some of the things. What I would like to first mention is that there were 33 centers that took part in MUKeight across the UK, and that randomization was carried out by an automated 24-hour web-based system managed by the clinical trial unit in Leeds to balance treatment groups for age under 60, 60 to 69 versus 70 plus years, the number of prior lines of therapy, up to 3 or more than 3, and beta-2 microglobulin from under 3.5 to 5.5 or more milligram per liter. Finally, during the trial, after a protocol amendment, what we introduced was the possibility of a crossover from CD to ICD upon disease progression. This was when ixazomib was approved by NICE to enable patients to have access to ixazomib. So in total, 112 patients were now back on the left-hand side of the slide in the flow diagram. 112 patients were assessed for eligibility. 58 were allocated to ICD and 54 to CD. And of those, because of a number of reasons, those you can look up if you're interested in the paper, 57 actually received ICD and 53 received CD and were also in the evaluable population. So let's look a little bit at patient characteristics and look at the minimization or randomization factors. First, if we look at age, about just under 20%. On the right-hand side, you can see the total for the trial because there was a very good balance between ICD and CD in general. I think you can just look at the total mostly here on the right-hand side of the table. Just under 20% of the patients were younger than 60 years, about a third were 60 to 69, and just over half the patients were actually 70 plus years old. So a relatively old patient population. In terms of the number of prior lines of therapy, 80% had more than 3 prior lines of therapy. And here you can see a relatively equal distribution across the 3 beta-2 microglobulin concentration ranges. If we go a little bit further down and look at characteristics in a different way, we can see that the median age of patients included was 70 years with a range from 46 to 82. The median number of prior lines of therapy was 4 with a range of 1 to 5 plus, so quite a bit. And just over 50% had up to 4 lines of treatment. Prior therapies, as you can see, almost 100% had either bortezomib or lenalidomide and thalidomide, and actually almost 20% had already been treated with a CD38 antibody, and just over half had undergone a prior stem cell transplant. The time since diagnosis is relatively long, almost 7 years. So you can see a quite heavily pre-treated, relatively old patient population. If we look at further characteristics, particularly here, the frailty score is what I've highlighted. And this is a simplified frailty score based on a frailty score established a couple of years ago by Thierry Fasson and since then used in a number of trials. This frailty score classifies patients into frail and non-frail and is based on age, the Charlson Comorbidity Index, and ECOG performance status. And what you can see here is that 73.6% of the patients in the trial were scored as frail. That is quite high. In other trials, for example, like the Boston trial, where this frailty score was also applied, only about a third of patients, up to 40%, maybe are usually qualified as frail. So to summarize, relatively old patients, 70 years old plus, 4 prior lines of therapy, and largely classified as frail. That is, I think, important to take with us as we start to look at the results. And here I want to start with the best response. In this table, in the red square, you can see the ICD results, and in the blue square, the CD results. And what you can see is that only 1 patient in each arm achieved a CR. In terms of VGPR, a slightly higher percentage of VGPR in the ICD arm, 14% versus 9.4%. PR, pretty much the same, 26-28%. Minimal responses, slightly more in the ICD arm, 15 versus 11. Stable disease or no change, less in ICD versus CD, but then again, slightly more progressive disease in ICD versus CD. So really, what we can see here is that there was no clear or definitive difference in response rates between ICD and CD, which is potentially already a bit of a surprise. Now here the main finding really, progression-free survival. In blue, CD with a median PFS of 6.7 months, and in red, ICD with a median PFS of 5.6 months. This was not statistically significant at all. So no difference here in PFS between ICD and CD. Looking at overall survival, maybe again a slightly unexpected finding in the sense that the median overall survival for patients in ICD was 14.1 months, whereas for those treated with CD, it was 19.1 months. Now, this wasn't statistically different, but it looked like there was a trend towards better overall survival in the CD arm. And I'll go into that a little bit more in the next slide. So here we've also tried to break down the PFS and overall survival data into whether patients were classified as frail or non-frail. And in the table at the top, you can see really that for PFS, there wasn't any sort of clear difference. But if we look at overall survival, in the non-frail ICD group, the overall survival was 15.3 months, 12.5 to 12.52. I think that the 3 is an error here we have to correct, that compared to not estimated in the CD arm. And the frail patients really in the ICD arm 14.1 months, 11.1 to 15.7, compared to 18 in the CD with 13 months to not estimated. And you can also see a little bit further down in the forest plot that it really looks like that there was almost or very close to a survival benefit for patients on CD, particularly for the frail patients. Now, why might that have been the case? Obviously, numbers are a bit small to really draw any definitive conclusions. If we look at the adverse reactions, we can maybe learn a little bit about this. Here again, on the left-hand side, you can see the ICD results. On the right-hand side, the CD results. And what I've highlighted is some of the main adverse events. If we first look at anemia and thrombocytopenia, particularly those of grade 3 or more, we can see there were considerably more anemia and thrombocytopenia events in ICD, with about 29 and 45% compared to 22 and 24%. If we go further down, we can see that neuropathy was somehow more common also in the ICD arm, as you would expect, particularly if you look at grade 3 or more. None of those were recorded in the CD arm, but 5.3 and 1.8% respectively for sensory and peripheral neuropathy in the ICD arm. Now, what's also important, I think, is to look at those modifications, omissions, and severe adverse events, because here we can clearly see a disadvantage of ICD. In terms of overall dose modifications, those were recorded for 71.9% in the ICD arm, compared to 54.7% in the CD arm. Those omissions also much more frequently observed in ICD patients, over 60%, compared to about 26% in the CD arm. And severe adverse events were also observed in 59% of ICD patients, compared to 49% in the CD arm. So it really seems like there was an issue in the ICD treatment arm with those modifications, dose omissions, toxicity. A few words about the CD to ICD crossover. As I said, this was introduced after a protocol amendment to allow patients access to ixazomib after NICE approval. A total of 25 patients were assessed for eligibility for the crossover. 21 were eligible, and about 20 actually received the ICD crossover treatment after progressing on CD. The median PFS from cycle 1, day 1 of ICD crossover was 4.6 months, with an overall response rate of 25%, 5 out of 20 patients, 3 achieved VGPR, 2 a PR. 50%, however, 10 of 20 did have stable disease. So it looks like there was some element of a clinical benefit of the crossover, although it was certainly not a major one. Adverse events and dose omissions reductions in the crossover ICD path were sort of comparable to the primary ICD arm. And with that, I would already like to come to a summary of the findings and an interpretation with it. So what we can clearly say is that ICD did not improve key outcomes compared to CD, and that certainly was not what we had expected. What's the explanation for this? One is probably that ICD seems to have been a little bit too toxic for the frail and heavily pretreated population included in this trial. The question is, were the issues that we observed with ICD specific for ixazomib? Probably, I would say not, because as we all know, ixazomib is in principle rather well tolerated in most patients. So it seems that we just have to be very careful if we evaluate triplet therapies in general, in frail, elderly, heavily pretreated patients. And that certainly in that patient group, it is not a given that a triplet is better than a doublet. Now, we observe this trend towards an inferior overall survival with ICD, particularly in the frail patients. While we don't have a definitive answer for that, I do believe that it has something to do with toxicity, possibly with the ability also to then have further treatment and tolerate that further treatment, although we don't have the data to really demonstrate that. And maybe the key finding to take away, that is in my view, not necessarily a negative one, it's a positive one, is the relatively favorable outcomes with cyclophosphamide and dexamethasone, with a PFS of more than 6 months, an overall survival of more than 19 months, in a really quite advanced and frail patient population. And what cyclophosphamide and dexamethasone is it's an all-oral combination, it's simple, it's really quite cheap. And I think at a time when we go to meetings and everybody is talking about bispecifics and CAR T cells and then these types of treatments, this result is obviously not a very sexy one. It's not a very attractive one, maybe necessarily, but it is an important one, I believe, particularly when we take a global outlook, because most of the world is far away from having access to modern, very expensive therapies. And for those, I think the news that cyclophosphamide and dexamethasone alone can actually have a pretty decent outcome, even in advanced and frail patients, is good news and helps support the choice to use this combination. And also, if we think about accessibility of care after 2 years of COVID restrictions, I think this is something to take into consideration. Also, when we think about geographical remoteness, frail patients, and how easy they may find it to get to secondary, tertiary care units. So cyclophosphamide and dexamethasone is, in my view and in my experience, a combination that is worth thinking about in certain patient groups. And with that, I would already like to thank you for your attention. Again, extend my thanks to all the centers that participated in the trial. And I will now hand back to Sarah for closing remarks and coordinating questions if there are any. Thank you.
Thank you so much, Holger. That was a great presentation. Really nice to see all of the results brought together and presented so very clearly for us. So thank you. If anyone has any questions, please do pop them in the chat or raise your hand. I will try to keep attention on the side panel with the participants. But just while people are thinking, I guess we've talked through the results a lot, Holger, obviously, whilst we were writing the paper. And one of the things that struck me as you were presenting was that you gave the background really nicely to us to why we were considering ixazomib in this particular combination, and the really positive results that we'd seen from the phase 2 trials. And clearly the population that we recruited within MUKeight was very different to the population that was in those previous trials. So I think we've got a median age of 70, whereas one of the previous studies, the median age was 63, much more heavily pretreated population. And I was wondering, do you think we need to do more background research, perhaps more extrapolation of previous data ahead of designing clinical trials in these more heavily pretreated elderly populations?
So I think and that's something that's obviously already being initiated also in the UK. We need to think about heavily pretreated frail patients in a different way than we do for the newly diagnosed or earlier relapses. And obviously all the attention is on these relatively young and fit patients, newly diagnosed, earlier relapses, those where the pharmaceutical industry also obviously prefers to do trials because the results will inevitably be better than if you go to fifth line treatment in 75 plus year old frail patients. Those will always be very difficult. But that's why I think it's important still that trials like these are done, because usually I think it's easy to list a long list of trials where 3 drugs gave better outcomes than 2. And this is one of very few where this wasn't the case. And it just highlights that more isn't always better. And in terms of should we do more background research, I think still this wasn't predictable. It sounded like a very good plan, and it really sounded like ixazomib in combination with cyclone dex should have improved outcomes compared to cyclophosphamide and dexamethasone. I still don't see how we could have predicted that it wouldn't. It's just that's why we do clinical trials. We really need to find out. We can't just say usually that's the case, so that'll be the case, let's do it. That's clinical trials.
Yeah, absolutely. That's why we do what we do. All right. Thank you for that. So we've got a question from Jane in the chat. So thank you for the presentation. Can we ask whether you think the dose of 500 milligrams of cyclophosphamide may have been too high, and could lower doses of cyclo reduce some of the toxicity that was seen?
Yes, and I'm sure there was a lot of dose reduction going on. I can't remember now how much we looked into the overall dose reductions. I know we have a result that I believe is in the supplementary of the paper that lists what exactly the cyclophosphamide dose, the median cyclophosphamide dose given was, and it was definitely below the maximum. I think in practice with treatments like this, we always adjust. And the adjustment of cyclophosphamide and dexamethasone was pretty much similar in both arms. And the ixazomib overall dose, if I remember correctly, was a median of 3.7. So those reductions were made. Could lower doses have made it more tolerable? Probably. Would that have changed the main outcomes? I'm not entirely sure it would. Does that answer the question, do you think, Sarah, or should I keep going?
Sorry, I think my internet connection has gone, so or is waning. So apologies.
Maybe it's mine.
I can hear you now, so hopefully it's
okay.
all right. Yes, I think that does answer the question. And as you say, we did look into the dose reductions of cyclophosphamide, and I also can't remember that off the top of my head, but it was certainly something that we looked into.
I also remember that the sort of treatment termination because only of toxicity was, I think, similar in both arms. So it wasn't a very clear picture that ICD was definitely way too toxic, but there were some indications like the dose omissions, like the SAEs, like the dose modifications that pointed to it, the toxicity being some problem. But we don't have a definitive answer, I think, to that.
Yes, I agree. Thank you. So Chris Parrish has got his hand up. Chris.
Thanks, Holger. That's a really clear explanation of the data. I really enjoyed that. It was more of a comment really than a question. I think one of the things that strikes me about this trial is that obviously the result is perhaps not the one that we were hoping for or even expecting, but in some ways I think that maybe part of the reason for that, as you've talked about, is that this was actually reflective of a much more real world population than we're actually used to seeing in trials like this. And to some extent, I think that's the trial is sort of a victim of its own success because it had this very, realistic recruitment strategy in terms of inclusion and exclusion criteria. And actually I think that's something that I know it's difficult, but that's actually something that we should be celebrating, that this is a trial which actually reflects the real world rather than the very heavily selected populations that we see in lots of the studies that are published at later lines of therapy. I know that's not what we want for a meeting or what the journals want, but actually I do think that's really important with this study.
Yeah, I completely agree with you, Chris. And I'm really delighted that actually Blood Cancer Journal decided to publish this, because I think they realized that it is an important finding, even though it certainly is not sexy. I don't have a better word for it. But it is important. And I think cyclodex having a PFS of close to 7 months is in a way encouraging in this very, very heavily pretreated patient group. And there was a real world data observation of pomalidomide cyclodex published a couple of years ago in less heavily treated patients. And the PFS there was only about 7 months. So that's, as you said, this is the real world. The real world is different from the flashy CAR T cell trials in that we may sometimes see highlighted in meetings. Yeah.
Yeah, I would wholly agree with that. And I think one of the when we opened the study, we were, I think there was little apprehension around the CD as a control arm, and whether, as you say, Holger, that was sexy enough for patients to want to be recruited to the trial. So actually the results are really positive from that angle, and it's good to be able to demonstrate that within this trial.
And I think it's important maybe to point out that I don't think it's a problem that's specific for ixazomib. I think if we had used a pomalidomide or I don't know, bortezomib in a similar trial setup, I would dare to speculate that the results wouldn't have been much different from what we saw here. I mean, there's obviously the possibility that ixazomib is slightly less effective than maybe bortezomib because of the different discussions around this. But overall, I think this just highlights that in a heavily pretreated real-world kind of population, slightly different rules apply.
Yes, very much so. Thanks, Holger, and thanks, Chris, for the question. So I'm just having a look through the list of participants. I can't see any more hands up, and there aren't any more questions in the chat. So unless anybody shouts up, I'd just like to take this opportunity, Holger, to say a huge thank you from myself and everyone who's joined the talk today. It's been really great to hear from you, and great to hear those results. And thank you to everybody for joining us. We are planning further seminars or webinars rather. We've yet to confirm the date of the next webinar, but we will be circulating that in due course. So please, if you are subscribed to our emails, please keep an eye out for future webinars from the CARP programme. So thanks very much, everyone. I hope you have a lovely rest of the day. It's almost the weekend, and I look forward to seeing many of you soon. Thank you very much.
Thank you. Thank you. Have a good afternoon.